Fluorouracil is what we'll be talking about today. Fluorouracil, or also known as 5-FU, is a chemotherapy drug that's been in use since the 1950s. It was developed after scientists noticed that cancer cells take up uracil more quickly than normal cells. Since uracil is one of the base units in RNA, 5-FU was designed to look like uracil but act like a decoy. When cancer cells absorbed 5-FU, they unknowingly activated it into toxic forms that interfere with DNA and RNA processes. Because cancer cells divide rapidly and rely heavily on both of these functions. 5-FU is especially effective in targeting them. Once inside the cell, FU doesn't act immediately. It must first be metabolized by several enzymes. These enzymes convert to 5-FU into three action forms: F-dump, FUTP, and FDUTP. Each one has a specific role in interfering with cancer cell survival. F-dump inhibits DNA synthesis by blocking an essential enzyme. FUTP gets incorporated into RNA, causing dysfunction in RNA-based processes like protein synthesis. FDUTP gets mistakenly added into DNA strands, which damages the genetic code and creates instability. This multi-pathway act makes 5-FU more effective and harder for cancer cells to resist. The metabolite FUTP is structurally similar to UTP, a natural building block of RNA. Because of this similarity, the cell's RNA polymerase mistakenly incorporates FUTP into RNA instead of UTP. This results in faulty RNA that cannot be translated properly into proteins, disruption of messenger RNA, ribosomal RNA, and transfer RNA affects everything from enzyme production of cell structure, since cancer cells need to constantly make proteins for rapid growth. Even a small disruption in RNA synthesis has a major impact on their survival. The most famous action of 5-FU comes from F-dump, which inhibits an enzyme called thymolate synthase. This enzyme is required for making thymine monophosphate, a precursor for the DNA base thymine. F-dump binds tightly to thymolate synthase and forms a stable inactive complex with the help of a folate cofactor. When DTMP production stops, the cell can no longer make the DNA it needs to replicate. This causes something called thymine-less death, which is lethal to rapidly dividing cancer cells that are preparing to duplicate their DNA. Another dangerous metabolite produced from 5-FU is FDUTP. It resembles DTTP, a natural DNA building block, and gets misincorporated into DNA during replication. But FDUTP doesn't pair correctly and creates unstable, mismatched DNA. The cell's repair system recognizes this damage and attempts to fix it, but constant misincorporation overwhelms the repair mechanisms, especially DNA strand breaks and replication stalls, leading to cell cycle arrest and programming cell death. This pathway works together with FDUMP's enzyme inhibition to maximize danger to the cancer cell's genome. 5-FU is rarely used along in modern treatment. It's typically combined with other drugs like leucovorin, which stabilizes the bond between FDUMP and thymidylate synthase, enhancing its inhibition. Its colorectal cancer 5-FU is part of regimens like FOLFOX and FOLFIR, which inhibit oxypathlate and irinotecan. These combinations attack cancer cells from multiple angles and have been shown to increase response rates by up to 50%. Because of these combinations, 5-FU remains a first-line treatment dedicated after it was first introduced, especially for colon, rectal, breast, and stomach cancer. In summary, 5-FU is a powerful anti-cancer agent that uses a three-pronged approach to destroy cancer cells. First, FDUMP blocks the synthesis of thymine, preventing DNA replication. Second, FDUTP is misincorporated into DNA, causing damage and genomic instability. Third, FUTP disrupts RNA synthase, stopping the cell from producing protein. Together, these mechanisms slow or completely halt cancer cell growth. By combining 5-FU with other drugs, clinicals can improve its potency and minimize the chances of resistance. Its targets actions on fast dividing cells make it an essential tool in cancer treatment.